Thank you. Hello, everyone, and thank you for being here. This is the first of three installments in the Beyond Mammals Exploring Cancer Models series, with this webinar being focused on the model organism C. elegans, the worm. I'm Justin Benavides, and I'm a program director at NCI's Division of Cancer Biology. We have a full agenda today, but first, a bit of housekeeping. We're going to open this webinar with three talks from our wonderful speakers, and each talk will be followed by a short Q&A that will be focused on the research presented during those short talks. Please save all of your questions until then. During the Q&A, feel free to put your questions in the chat and I can read them from there, or raise your hand so we can unmute you and you can ask your question verbally. Following the talks, we're going to have an extended discussion session. So I'm going to open the discussion with a couple of standard questions, and then I'll open the floor to attendees for you to ask your questions. Please take care and help us create an atmosphere of mutual respect and sensitivity during this discussion. You know, we're all friends here and here to talk science. And finally, I'll say a few words about this series. Here at the Division of Cancer Biology, we have a powerful interest in cancer models. Much of the research funded by our division is in mammalian models that we all know and love, like the mouse, and a growing proportion is dedicated to developing computational models for cancer. But the model landscape is wider than just those two categories, and in this series, we hope to highlight the work of researchers who use non-mammalian animal models like the worm to answer cancer biology questions and start a conversation about cancer modeling more broadly. So without further ado, I would like to introduce Dr. Dave Reiner, professor at the Center for Translational Cancer Research at Texas A&M, Dr. David Matus, researcher at UC Berkeley, and Dr. Swathi Arur, who is professor at MD Anderson Cancer Center. First, we're going to see a presentation from Dr. Reiner. So I'm going to hand it off to Dr. Reiner. Go for it. Let me just share your slides.
All right, thank you, Justin. Thanks for organizing. Thank you, NCI, for organizing. Thank you for handling my slides. So I'm actually located in Houston. I could get to Swathi's office in about a speedy 10-minute walk. So we're neighbors and see each other regularly. That's great. Are we all set?
Yeah, right. Good to go.
Okay, so today I'm going to talk to you a little bit about a poorly understood RAS oncogenic signaling pathway and how it intersects with the exocyst complex in C. elegans. I've shown, and I can't use my highlighter, so this is tough. But I've shown my former grad student, now postdoc, Yo Wu, who has generated all the worm data in this talk. Next slide, please. And I was asked to actually recap briefly the strengths of the C. elegans system. So I'll go through these for those of you who aren't familiar with them. So short generation time and ease of culture, great genetics, great transgenics, and you can do them very rapidly. Little or no paralog redundancy and conserved human disease genes. This is critically important and important for this talk as well. They're optically clear, so we can easily image in live animals. And there's a defined cell lineage. Nearly every somatic cell is in the same time and place. I added somatic for Swathi's benefit. I'd get in trouble if I hadn't done that because she's a germline researcher. As relevant benchmarks to give people some context, just arbitrarily, but Nobel's as high impact discoveries. So apoptosis in the EGF-RAS MAP kinase pathway are consequences of that cell lineage and was one of the earliest major advances in C. elegans. The first animal to express GFP transgenically, RNAi, we can actually feed them RNAi, which is tremendously helpful. And of course, most of you are probably familiar that last year's Nobel in microRNA. So they're major discovery advances. I'm going to put in a plug for Cynthia Kenyon on the genetic determinants of lifespan, which I think was also a major advance. And so one key concept here is that of model discipline. And I want to emphasize that in this context, it was originally, I was familiar with the concept, but the term came from a conversation with a seminar invitee several years ago, Robin Tanguay. And I love the term model discipline. Knowing what your system is great at and knowing what it's not great at and designing your questions and your experiments appropriately. Next slide, please. And so today I'm going to give you some background on the signals we study, and we finally, I think we decided to tackle the 800-pound gorilla or elephant in the room, however you want to put it, the exocyst complex. Next slide, please. And the exocyst, this starts with RAS. So in a cancer group, almost everybody's familiar with RAS. It's the most mutated oncoprotein. It is on in the GTP bound state. It cycles to the GDP bound state, which is off, and that's regulated by GEFs and GAPs. And that this is important for downstream of RAS, which is RAL. RAL stands for RAS-like, and it's also a small GTPase. Next slide, please. And so everybody in the cancer group is probably familiar with RAS-RAF and RAS-PI3 kinases, among the most studied and pharmacologically targeted proteins in all of biology. But a neglected sibling is the RAS-RALGAF-RAL proto-oncogenic pathway. This is probably neglected because it signals to the exocyst complex. And I'm going to talk about that, but that seems to have been a dead end for sort of typical biochemical bootstrapping and understanding what's going on downstream. And so that's where we come in, because we're going to use the strengths of C. elegans genetics to address this, plus some modern advances. Next slide, please. And so what is the exocyst? So the exocyst came out of Randy Sheckman's original screens for secretion defective yeast. The exocyst seems to escort the direct secretory vesicles from the Golgi to the plasma membrane to tether them for snare-mediated vesicle fusion. Yeast, however, do not have RAL. They have a Rab-like protein called SEC4, and it seems that in animals, RAL has substituted that. And so that represents our puzzle. We have an important signaling molecule in oncogene that's actually participating in a central and essential cell biological process. Next slide, please. Thank you. And so this is a gorgeous cryo-EM published several years ago, the yeast exorcist. Next slide, please. And on top of that was the discovery over 2 decades ago now that RAL interacts, this is in cell-based systems, RAL interacts with the XO84 and SEC5 of the exocyst and seems to govern cancer-like behaviors in those cells. Next slide, please. So our puzzle then is that RAL is likely essential for exocyst function in animals, but most of the data are circumstantial. We think that RAL binds and employs SEC5 and XO84 as signaling intermediaries, but the gap here is that any genetic perturbations or pharmacological perturbations, for that matter, of RAL perturbs both signaling and the exocyst complex, and the exocyst is required for cell viability. So, secretion is important. It won't surprise you to hear that. Next slide, please. Okay, so I'm going to briefly give you a history of our work in C. elegans signaling, and then that will point very quickly to our exocyst studies. Next slide, please. So we have studied signaling via XO84 in the vulval precursor cells, upper left. RAS actually switches effectors between RAS-RAF and RAS-RALGAF-RAL in equipotent cells. That was a pretty interesting phenomenon to begin with, and then we populated a pathway downstream of that through XO84, a MAP4 kinase, and P38. RALGAF itself is multifunctional. In addition to its RAS-RALGAF-RAL function, it scaffolds PDK, AKT, and we've got a mystery function we don't understand when we delete the whole protein. So we're very careful about how we mutate that. RAL regulates lipid storage, RAS-RALGAF-RAL, and that's in the center. RAS-RALGAF-RAL mediates cell migrations, that's the lower right. And now, over a decade ago, we published a RAL connection to mTOR in cancer biology. Most of that paper is cancer biologist in Channing Durr's lab, and I'm specifically going to give a shout out to Tim Martin, who's now an assistant professor at UVA, who spearheaded all of that cancer research. There's really only one worm figure there. Next slide, please. And we got the cover. Just very happy about that. So RAL is an essential regulator of the exocyst complex. That's our conclusion, and we wanted to show A, that it's actually regulating exocytosis, and B, that it physically interacts. Next slide, please. And this is a laundry list of all the things we checked off. It's very daunting. Genetics, various assays for exocyst function, cargo transport, cell autonomously versus non-autonomously, even mutating the interface of SEC5 and XO84 based on structures, co-localization, and immunoprecipitation. Next slide, please. We'll only touch on a couple of these. SEC5 and RAL mutants are actually maternally rescued, which means that their moms are heterozygote and load a lot of gene product into the embryo, and they make it to adulthood. They're sterile and not entirely happy at that point, but if we deplete all of it, they die as embryos or early larvae. And the double mutant shown here, the healthy worm is a double heterozygote, the insect is a dead embryo that's a double homozygote. They never hatch. And so we performed a wealth of genetic studies to address these interactions. Next slide, please. One of these is the PVD neuron arborization, shown here are single mutants and the double mutants. So as you decrease exocyst function via mutant combinations, you get a decrease in arborization of these neurons, which had already been established to be dependent both on exocyst and recycling endosomes. Next slide, please. And those data are quantified here. The exocyst XO8 RAL double is the worst. We don't show SEC5 RAL because they're dead. They don't make it this far. Next slide, please. And pretty pictures, because I couldn't resist. Yo made such beautiful assays for trafficking, et cetera. So here's a pretty picture slide. Next slide, please. Also gorgeous picture. Next slide, please. No data, just to show you how awesome her data are. This is probably the smoking gun that we tagged all these. We did co-localization. These are endogenous proteins, I should add. Everything's endogenous, and mutants, tags, fluorescent probes, epitope tags, etc. Okay, so these are co-IPs, reciprocal, and we've done it every which way, which is basically SEC5 and XO84 interact. SEC5 interacts with RAL, XO84 interacts with RAL. Next slide, please. Okay, so that summarizes the laundry list, is that if it walks like a duck and quacks like a duck, it's a duck. And RAL is actually performing exocyst-like functions and is actually interacting with exocyst components. Next slide, please. So now, with our system established and having finally proven this in a live animal rather than, 20-some-year-old studies in cultured cells with the limitations of the time, right? It would be much better now. We can get on to asking some major functions of how RAL interacts with the exocyst and how this process is regulated. So the first of these we found is a signaling deficient RAL mutant. Next slide, please. And this is graphically illustrated here. These are the 4 points I'm going to make. So this first one, this mutant RAL, it's a point mutant in this sense. It cannot signal, but exocytosis is fine. Next slide, please. So this was discovered serendipitously by the Million Mutation Project. This is an alignment of all the human and all the elegans RAS proteins with a couple outliers thrown out, but this is the basic RAS family. This arginine is conserved. It's right next to the G4 box. And this R139H is defective in all of our signaling assays. Those include a couple studies of the equipotent vulva precursor cells shown in the middle and our cell migration assay shown on the right. Next slide, please. And this mutation disrupts the salt bridge. I actually found discussions of the salt bridge in some of the original RAS structure papers. And every alignment I look at and every structure I can find of small GTPases has this. This includes the Rho family and the RAB family. So it seems to be broadly functional, at least as a structural element, but these are actually perfectly live animals when we mutate this, and RAL's an essential gene. So our first step was to CRISPR these into wild type animals to get rid of the caveat of heavily mutagenized backgrounds. The mutations do not affect protein stability. This includes the original R139H and mutating the other side of the putative salt bridge, E159D. Next slide, please. And these are phenotypic assays. It would take me 5 or 10 minutes just to set this up in vulva development, cell migration, et cetera. But the idea is the original mutation is called GK62881. Our CRISPR alleles get their actual amino acid change names. And you can see they're defective in the same way as the original mutation, left, center, and right is our cell migration assays. And so these are signaling defective indeed. We validated it this way. Next slide, please. Importantly, I mentioned before that deletion of RAL in Sec5, that would be column 5, is lethal. Mostly dead embryos, a few survive to hatch. But the signaling defective RAL, which is normally completely wild type and by surface or superficial inspection, actually causes no increased defect in exocytosis, even in the most sensitive reduced function background. So we don't think that this compromises exocyst function at all. Next slide, please. So our hypothesis, or my hypothesis, was that this was a GDP-bound protein, right? And I thought, oh, okay, and the reason is that salt bridge looks like it's holding the G4 and G5 boxes together like a pair of forceps. And G4 and G5 actually grasp the purine ring of guanine nucleotides, very important for small GTPases, right? And so I thought, well, we're relaxing that and it's going to lose specificity. This is probably GDP bound or nucleotide free. And yet this is actually, this protein, or the human equivalent, the mutant loads GTP at the same rate as, not significantly different from RALA in humans. This was done by Matt Smith's lab at Montreal. So it really changed our thinking. These days now I'm wondering, okay, maybe this is affecting interaction with the GEF, or maybe this is something in dimerization. And I know dimerization is controversial in the RAS world, but maybe this mutation is actually, I don't believe in obligate dimerization of small GTPases, but permissive dimerization where they're forced together by membranes or effectors could be the case. So these are both things we're thinking about right now. Next slide, please. And so the next story will be pretty brief, is that we think GDP-RAL is actually sufficient to function in the exocyst. That is, you don't need upstream activation. It's just there as an anchor. Next slide, please. And this is simply because the GEFs are non-essential and RAL itself is essential. So shown on the left, we actually have about 7 alleles of the GEFs, including a deletion that takes out 9 genes, including the GEF. And they're perfectly happy animals. You can see that they're signaling defective in the right backgrounds. When we activate the signal, it'll block it. But RAL itself is essential, as I discussed. We also characterized a RAL-GEF signaling defective, just a conserved residue in the RAL-GEF that will block in our signaling assays, but does not enhance the exocyst defect. So we have now got in the lab on plates worms. We just CRISPRed in the canonical catalytic dead RAL-GEF, so the catalytic arginine to glutamate change, and so that's a more understood reagent, but this is still a puzzle. It's not affecting exocysts at all. It's just blocking signaling. Next slide, please. And our goal here is to really test this. We can't really do this mutationally, so we're expressing transgenically a constitutively active gap. And so there's a specific RAL gap. We're driving the gap domain using RAL membrane targeting sequences shown in the endogenous RAL here we published a few years ago. And we think this will drive the RAL protein entirely or mostly to GDP-bound form. We expect that this will block signaling but not exocytosis. So that's our hypothesis. We are not impervious to the notion that there are other GEFs out there, and that explains our results. They don't appear to be required for signaling, and there are none by homology that would be good candidates, but that is our backup hypothesis. Next slide, please. And so the last one here is, if RAL signaling is not required for exocytosis, what happens with elevated signaling in the RAS, RAL-GEF, RAL pathway? And we found that it actually promotes exocyst-dependent exocytosis. Next slide, please. And that's shown here, a little complicated, has no effect on normal animals. In column 5, the yellow column is the XO84 Sec5 double. Those die as early larvae. They're deficient. They're not absent exocyst, but it's low. Activated RAL will bump that back up to sterile adults. So they live literally 24 hours longer. It's an almost doubling of their lifespan. And of course, those animals survive just fine for their full lifespan. They're just sterile. And importantly, the weaker activating RAS mutation will also reverse that early lethality. So we think the RAS-RAL-GEF-RAL pathway is promoting that. Down below, rather than viability, we're seeing, C versus D. This is the arborization of little neurons, our assay. RAL-GEF increases the little hair-like structures, which are the quaternary dendrites. In an XO84 background, SEC5 background, few dendrites, RAL increases that as well. So next slide, please. And this is probably the most important is our co-IP is much more robust in the activated RAL background. So it seems that GTP-bound RAL, constitutively active, has much higher affinity or much more of the protein is binding to the exocyst. That's compared to the wild type, which is in where you should be looking at the third three lanes and not the first three, which are input. This is an IP of XO84 pulling down RAL, and we see much more with activated RAL compared to the wild type. I should add our signaling defective RAL doesn't change at all relative to the wild type. Next slide, please. So we think this points, and one other thing we're trying to do, since the endogenous RAS mutation is weak, and we can't, we made a G12V RAS, which is the fully activated, it kills the worms. So we're doing transgenic RAL-GEF selective activated RAS to try to drive this pathway in a way that doesn't activate other signals just through RAL-GEF. That should not be lethal. We used this in our vulva studies before. And so we're hoping that that will boost exocysts to the point where we can actually do our IP with RAL again. Next slide, please. And so then that leads us to the last model. Like if RAL just anchors the exocyst, but activated RAS and RAL is an input, maybe from the whole animal to upregulate exocytosis, then maybe that's actually the oncogenic function of RAS-RAL-GEF-RAL is exocytosis. So let's look at that last question. This is a collaboration. Next slide, please. It's a collaboration with Chris Counter at Duke. This is standard to the cancer biologists where you deplete RAL-A, in this case, you rescue with wild type RAL-A or signaling mutant RAL-A. Next slide, please. These are the results in colony formation. Next slide, please. And this is the quantification. And I just highlight two of these, which either the wild type RAL-A or the RAL-A signaling defective will rescue the colony formation. Next slide, please. And proliferation, same general set of genotypes. Next slide, please. And so for me, this actually suggests then that the RAS-RAL-GEF-RAL pathway is not promoting oncogenesis through some signaling activity like has been assumed from the get-go, that it's actually exocytosis. And how would that work? The idea would be RAL-A and RAL-B are thought to govern different aspects of oncogenesis. Perhaps they're trafficking exocyst complexes to different compartments. Perhaps as an idea, maybe apical versus basal or something like that. Obviously, it could be much more complicated in a cell in a live animal. Next slide, please. All right, so we've hit the major points. We've established this model. And we've made four independent, well, not entirely independent, we use the same reagents, but four new observations about how the RAL and the RAS-RAL-GEF-RAL pathway might intersect with the exocyst functionally, and trying to get at some mechanisms of how this might work. And our long-term goal, this is a proto-oncogenic pathway, so we're interested in how this might impact cancer. Next slide, please. Wanted to thank all the folks in the lab and collaborators shown with the purple header there. Classic usual suspects with sharing reagents. And all the worm work here was done by Yo Wu. This includes CRISPR tagging of endogenous proteins and selective mutations and the whole bit. So quite industrious, and I think her joy of science shows here. She's also the official lab selfie photographer. Last slide, please. Thanks, everybody. I'll take questions here or at the end. I think I went over. I'm not used to doing it this way, but I hope that worked okay. Thank you, Justin, for very prompt slide control.
My pleasure. We probably have time for one question. If anybody has something, feel free to put it in the chat or raise your hand and we can unmute you. And if not, we can move on. Okay. That is all good. We can save questions for our discussion later. So thanks, Dr. Reiner. And next, we have our second Dave, Dr. Dave Matus. Dave, take it away.
All right, hopefully everybody can hear me okay. Great, so I'm really excited to be here today. Thanks, Justin, for setting up this really awesome series of sections. So all this work was done when I was a PI at Stony Brook University. After a brief stint in biotech, I'm happy to say I'm back in academia as a recently research faculty at UC Berkeley in the MCB department. And so I just want to start sort of in a similar vein as Dave Reiner did, that I think it's really important that we match our research question to our research system. And I encourage everybody to think really broadly that if you have a question, there's probably the right system out there to address that question, or multiple systems. And I think we need to avoid sort of square peg round holing our research into a system that maybe isn't exactly right for it. And so what I hope to convince you over the next 10 minutes or so is that we have a really, really great system to understand how cells invade through a basement membrane that just happens to be in C. elegans. And so with many of the strengths that Dave Reiner mentioned at the beginning of his talk, C. elegans is a really great system for trying to understand the cell biological basis of development. And so there happens to be a single cell in the developing larval C. elegans that adopts an invasive fate, and it does that on a clock. So every single worm, we know exactly when it's going to happen based on the division of these underlying global precursor cells that Dave Reiner works on. And the real beautiful thing is that we can watch this in real time using high resolution spinning disc confocal microscopy at multiple orientations. And I really challenge you to find any other system where you have this level of resolution with just a single cell that has to get through a basement membrane. And so I just want to give a shout out to my postdoc mentor, Dave Sherwood. I started this work when I joined his lab now many years ago. He really has dove deep into the cell biology of cell invasion. And so I just encourage you to go to his lab website and check out his research. This is from a recent paper. I think fundamental discoveries in our understanding of the cell biological basis of cell invasion are still coming out of his lab, and it's really exciting. When I was a postdoc in Dave's lab, I took advantage of one of the many strengths of C. elegans as a genetic model organism, and that is that I did lots of screens. And so I did two main reverse genetic screens using RNAi. As Dave Reiner mentioned, we can just plate out double-stranded RNA in bacteria and feed our worms. And so this enables us to do medium to high throughput screens. In this case, I took advantage of the fact that we knew many RNAi depletion phenotypes gave a defect in uterine vulval development that could be a result of the anchor cell failing to invade, because what I didn't tell you before is that the anchor cell's job is to connect the developing uterine and vulval tissues that a worm's can eventually lay eggs. And when that doesn't happen, you get egg-laying defects such as this protruded vulva. And so we knew a bunch of genes that caused protruded vulval phenotypes, or PVULs. And so we just looked at all of those at high resolution, just using DIC. So this is a DIC screen. So we don't even need any transgenes. We can see that the basement membrane is intact, and this would be a defect in invasion. And so that screen that I did at the beginning of my postdoc in the mid 2000s identified about 100 genes that were required for anchor cell invasion. And the really exciting part is about most of them, so 95% of them, had never been, the human homologs had never been correlated with cancer in any sort of way. And so I went to Steve Weiss's lab at Ann Arbor, and we just picked some of the top hits from the screen and just knocked them down in a couple of different cancer cell lines using chicken CAM assays. So there's a real basement membrane these cancer cells have to go through just to show that they were required for invasion across multiple cancer cell types. I hope that illustrates that we can go directly from observations in C. elegans to cancer experiments. And so the second screen I did in my lab, did in Dave Sherwood's lab that I took with me to start my lab, was I was really interested in trying to understand the gene regulatory network of anchor cell invasion. And so we built a screen where only the anchor cell and its neighboring cells were sensitive to RNAi. And this way, we were able to screen through most of the transcription factors that are encoded in the C. elegans genome. There's about 1,000 in the genome. We could target about 850 of them by RNAi. And this screen we did in high throughput, just looking for that protruded vulval phenotype. And so we took our hit list, and at the top of the hit list was a transcription factor that I ended up spending a lot of years working on. This is a nuclear hormone receptor. It's called NHR67, where names are pretty terrible, but it's a homolog of TLX or NR2E1 in humans. And on the left here is a wild type anchor cell. There's only one anchor cell, and it's invasive. And when you knock down NHR67 or TLX by RNAi, you get multiple anchor cells, so they inappropriately proliferate. And if you look with the basement membrane marker, this is a laminin transgene, you see that these multiple anchor cells don't invade. And so what I started my lab on at Stony Brook was really this dichotomy between invasion and proliferation. And so this transcription factor we found transcriptionally regulated one of the homologs of P21 or P27, a cyclin-dependent kinase inhibitors. These are tumor suppressor genes. And this circuit was required for the anchor cell to stay in G1 or G0 and important for differentiating the invasive fate so that when you lose the transcription factor, you inappropriately enter the cell cycle and you don't invade. And so I think, we all can pretty much agree that cancer in many ways is a recapitulation of embryonic programs. And so the question then is what we're seeing in the anchor cell also true in cancer cells? And if you sort of think about it, are cancer cells sort of able to like pat their head and rub their tummy at the same time? And so you can imagine there's many aspects during the metastatic cascade where tumor cells have to breach ECM or basement membrane. And the question is, do they need to be out of cell cycle when they need to do this? And so after we published our paper, we got asked to write a review on this because there actually wasn't one. And so it was really fun to dig through the literature with my first student, Abe Harmon, to see what sort of evidence was out there, something I had never thought of as a developmental biologist. And so there's a surprising amount of evidence. And so from immunohistochemistry showing a loss of proliferative markers at the invasive front of many tumor cell types, as well as at a cell biological level. So many cancer cells make invatopodia or invatosomes. These are punctate F actin-rich structures used to breach basement membrane. And people have shown in breast cancer cell lines that these structures are made primarily in G1. And the level of conservation is really striking. A couple of years ago, a student in my lab, Jay Smith, did RNAi screen identifying the epigenetic regulator, the chromatin regulator, SWI/SNF, as sort of a clear upstream regulator of the transcription factors that were controlling this dichotomy. And it turns out that in melanoma, this same sort of program is used where SWI/SNF controls levels of the transcription factor MIT-F, and MIT-F acts as a rheostat to basically, based on MIT-F activity, you're either proliferative or invasive. And so you can see that clearly tumor cells also need to make these decisions between invasion and proliferation. And so I was fortunate to have a really fantastic MD PhD student in the lab, Mike Martinez. And so for his thesis work, he wanted to ask the question, what is the role of the cell cycle control mechanism specifically, so independent of the transcriptional circuit that our lab was working on, in regulating this process? And so thanks, really lovely work in model systems such as Drosophila and C. elegans. A lot of this work was done by Laura Batista originally when she was a postdoc in the Edgar lab and then in her own lab at Ann Arbor. We know that the way cells exit the cell cycle during development is through a redundant mechanism. So not surprisingly, it's really important to exit at the right time and not to inappropriately re-enter. And so there are multiple cell cycle control proteins that need to be on to maintain a robust cell cycle exit, and that these need to be lost redundantly in order to trigger cell cycle entry so that you can activate your cyclin D and E complexes, which will lead to entering the cell cycle. And so I need to take one sidestep here and say that in order to do these experiments, we needed to make some new tools. And I think that has sort of been the theme of my career since I switched to worms, that I really, really enjoyed making tools and making tools for the community. And that's something I'm actively doing in my new role at Cal as well. And so as soon as I got interested in the cell cycle, we asked, well, what are the tools to actually visualize the cell cycle live? Because we wanted to be able to see not just the number of cells, but know exactly what cell cycle state cells were in. And so one of the first, I guess, major projects that my lab worked on was building a new cell cycle sensor in C. elegans, taking clues from in vitro mammalian systems and moving biosensors over to worms and zebrafish. And so what we decided on was using this kinase translocation receptor biosensor that reads out CDK activity. And so the way these biosensors work is that they have an NLS and an NES. And as your favorite kinase phosphorylates the serine sites around the NLS, they expose the weaker nuclear export signal. And so as kinase activity goes up, your biosensor goes from the nucleus to the cytosol. And so for a CDK biosensor, that means when a cell has strong nuclear signal, it's in G1 or G0, equivalent in S phase. And then when it's localized to the cytosol, it's in G2 or M. And so you can see in what these wild type invasive anchor cells stay with strong nuclear signal, so low CDK activity in G0 the whole time. And so Mike went ahead and crossed these beautiful reporters into single mutants of all these negative regulators of the cell cycle. And these are either null mutants or strong hypomorphs, and put the biosensor in there as well. And so what we saw is that loss of single mutants had no effect on the system. All these anchor cells invaded just fine and stayed in this G0 pro-invasive state. So the next thing Mike did was deplete that P21, P27 homolog by RNAi. And he found when he did that he saw a different result, that this time about 25% of the time, 1 out of every 4 animals showed mitotic ACs that didn't invade. And so given what we knew about redundant control of the negative regulators from other model systems, Mike just depleted CKI1 by RNAi on top of those single mutants. And what he found in that case is that he robustly triggered cell cycle entry in all of these backgrounds. And so what are the role of the cell cycle regulators? What we think is happening is that when you deplete these other regulators by themselves, we get compensation by CKI1. People have shown this in other systems, so we think that's likely what's happening. What's happening is that levels of CKI1 are going up to keep the AC in this G0 invasive state. And so it's only by hitting redundant regulators that we robustly trigger cell division. Now, if I know I was going really fast, so you might not have seen it. In some ways, I didn't want to talk about it yet. But if you notice in all these images that we see multiple ACs and breaches in the basement membrane. This was really surprising when these started to crop up in our imaging. And so when we're thinking about this, that really gave us 2 possibilities. One, it could be that the AC makes the hole in the basement membrane, then inappropriately enters the cell cycle and starts dividing. And so maybe the only a window in G1 in which to invade, in this case, then this invasion and proliferation dichotomy would be intact. The other possibility is that loss of cell cycle regulators just disrupts this whole system. And now the ACs are good at basically patting their tummy and rubbing their head, something I'm not great at. And that they can invade during any cell cycle state. And so this brings me to, I was in New York for 8 years, so I guess that means I get to use Yogi Berra quotes. There's a quote for everything. If you want a pen, my friend Paul Maddox says that you can really observe a lot just by watching in a microscope. And so the only way we can get this answer is using the strength of worms, is doing live imaging. And so we just imaged a bunch of worms where we started with a case that there was already 2 anchor cells. So we knew that the anchor cells were already proliferative. And then we just imaged. And so I apologize, the signal to noise is a little grainy in these movies, but I've annotated them. Hopefully you can get the point. So we've got these 2 anchor cells. We're imaging over time. And what we see in our CDK sensor is that the sensor is no longer nuclear localized. And so these ACs are in probably G2 when we first see this basement membrane gap. And so that means that cycling ACs, when we specifically target the cell cycle, can breach at any cell cycle state, breaking our dichotomy. And so what I didn't tell you before is that when we lose the transcription factor that controls this process, we lose some of these downstream effectors of invasion. So we lose the expression of MMPs or matrix metalloproteinases, and we lose the localization of the dynamic F-actin cytoskeleton, the invatopodia or invadosomes that are important for breaching that basement membrane. So we looked at those, and so sure enough, when we target the cell cycle specifically and have invasive mitotic ACs, they still make MMPs. They're a little bit downregulated, but they're still being made. This is a transcriptional reporter for one of the MMPs that's made in the anchor cell. And they still make these invatosomes or invatopodia. And so what's the difference in these two contexts? And the key one is that we haven't targeted the transcriptional pathway or the gene regulatory network, only the cell cycle in this context. And so we wanted to look at NHR67 as sort of the top of our transcriptional cascade. And so what we saw when we knocked in MD on green to the NHR67 locus of CRISPR in our mutants, that sure enough, the NHR67 still stays on in these mitotic ACs. So here's an anchor cell that's probably in S phase, still robustly expressing NHR67, and they're both invasive. And so I'll just end with our model here, is that basically, normally you have NHR67, which maintains the AC in a G0 pro-invasive state, limiting cell division. When we lose NHR67, we robustly enter the cell cycle, and we lose the transcriptional program that's important for pro-invasive gene expression. When we lose this set of negative cell cycle regulators, we get compensation by CKI1, which seems to be the key negative regulator of the cell cycle in this pathway. When we lose both, we trigger cell cycle entry, but we maintain the gene regulatory network that's required for invasion. So some of these ACs maintain their competency to invade. And so again, then taking sort of a step back and thinking about cancer as a recapitulation of development and all these contexts where cancers need to be invasive. I told you in the beginning, there's certainly evidence that this is happening in cancer, but of course, if you just PubMed invasion and proliferation and tumor cells, you'll get like 30,000 references that say that these are often hand in hand, and that genes that regulate invasion also regulate proliferation. So clearly, tumor cells are capable of, in some contexts, doing both these things. And what I think is really excited, this review couldn't be more timely. This came out last week in PLOS Biology. Very sort of bold statement about the end of the genetic paradigm of cancer. The thing I really like about this review is it talks about many of the things that are near and dear to my heart, and that really thinking about cells in their sort of transcriptional state and a combination of like cell fate and in Waddington's landscape, how cells have decision-making points that are based on both intrinsic and extrinsic cues that sort of guide these processes, and that like there are hills and valleys of these states. And you can imagine just by tweaking pathways that this could lead to situations where tumor cells can break out of traditional dichotomies like invasion or proliferation, so they may, in those contexts, be able to do both. And so I just want to end by encouraging people, again, to find the right model organism. And I didn't play this full GIF. I think if you have the right model organism, the right sets of models, you can have really creative solutions to your scientific questions. And I would encourage everybody to think out of the box like this really cute toddler. And with that, lots of people to thank. So Mike did all the work on the cell cycle, but I had a really great set of grad students and postdocs that helped me get through tenure and my transition out of Stony Brook through the years. And thanks for funding agencies, and I'd be happy to take any questions.
Thank you, Dave. Do we have any questions for Dr. Matus? Feel free to raise your hand if so, or you can type your question into the chat. Probably have time for one or two questions. I have a quick question maybe you can answer, which maybe about the cycling ACs that did actually manage to breach the basement membrane. In cancer, some cancers go through a process called collective migration when they're metastasizing. And sometimes, I guess I'm wondering about whether the potential to break through that basement membrane may differ between the different ACs in that context, and whether they may have potentially like different developmental stages or like mixed fates or something like that.
Yeah, I mean, I think that would be a great, I think like this would be a great model to model that, right? Like it would be really fun to have like your sort of tumor, AC tumor, and really sort of dig in at single cell level. Because I think in collective tumor invasion. So there are clearly leader cells, right, in those models. And I've always wanted to image cell cycle in those models to see if those tumor cells have any sort of cell cycle dependency, or if maybe there's switching going on and things like that. And if, or maybe they've just broken that dichotomy. I mean, I think like we just need to look.
Right. Yeah, super interesting question. All right, if we don't have any more questions from the audience, thank you, Dave. And we can go ahead and move on to Dr. Swathi Arur from MD Anderson. Swathi, take it away.
Okay, can you see my screen? Because I can't see it.
I can see it, but it's not in presenter mode.
Okay, perfect. All right, are we on?
We are
Yes.
on, yeah.
Perfect. Okay, thank you, Justin, for organizing this webinar, and my wonderful colleagues, Dave, Reiner, and Matus, for sharing your discoveries and for illustrating the power of how we can use C. elegans for making molecular and cellular insights. So the story I'm going to share with you today begins with C. elegans, and which we use to study oocyte development. So we use C. elegans as a model for studying female fertility and oocyte development. And I'll share with you how some of the work that we did in C. elegans to study these beautiful oocytes here led to identification of a potential biomarker that we can use to detect cancers with metastatic potential in humans. So in humans, by the time a human female is born, all of her oocytes are set aside. So all oocytes are already specified in utero in this human female child. And by the time she's born, they're arrested in meiosis 1. And this arrest in meiosis 1 stays pretty much through her childhood and into as she's growing into her young adulthood until puberty. And it's in puberty that the oocytes exit meiosis 1 arrest and undergo meiotic maturation and then arrest again for most of her reproductive life until the presence of sperm when they can get fertilized and undergo embryogenesis. So events that basically, that specify the formation of oocytes and how those events are coordinated with fertilization are questions that my lab has been wanting to understand for many years now. So everything we've been trying to do is trying to really understand how female oocytes are born and how they're coordinated with oocyte to embryo transition. As you can imagine, this particular question is a little challenging to study in humans or even mammals. But this is where worms become a fantastic model system to study it. So in worms have these two beautiful gonad arms right in the middle here, schematized with colors, and these yellow cells here are the oocytes. And if I zoom into this worm, what's really interesting is these oocytes are born continuously throughout adulthood. So here you have a steady state pool of germline stem cells that continuously feed the growing germline. These differentiate into myocytes or germ cells that go on to form arrested oocytes. These meiosis 1 arrested oocytes are very similar to the arrested oocytes that you see in humans, except that because worms are hermaphroditic, they receive a constant sperm signal in adulthood. And so every 20 minutes, once this worm is an adult, we can visualize events of how an oocyte exits its meiosis 1 arrest, enters meiosis 2, goes through fertilization. So in 1 hour, you can watch 3 of these events. And because it's C. elegans, we not only have dynamic control of how these events occur in a temporal fashion, we can also watch the signaling molecules that actually regulate the generation and formation of these oocytes. And the signaling molecule that I've been interested in my lab studies over the last 15 years is the ERK signaling molecule. So active ERK is a CD3 kinase that's phosphorylated downstream to a cascade of signals through KRAS. And you see this very striking expression of ERK, active ERK in 2 zones, in meiosis 1 and meiosis 2. I should note that total ERK is everywhere in this germline, but it's the active form of ERK that really matters and controls the maintenance and formation of these oocytes as they enter meiosis 1, become meiosis 1 oocytes, arrest, and then exit meiosis 1. And we can really open up these animals and get to fine cellular details. So here I'm showing you a dissected C. elegans gonad, which we use routinely in the lab. Your stem cells will be here. In blue, you're looking at the nuclei for each of these. And what you start to notice is here we've done a staining with active diphosphorylated form of ERK, and you see these two gorgeous expression patterns, which was schematized in the slide before, where you see active forms in zone 1 and zone 2. So to determine how ERK coordinates the formation of oocytes with maturation and then formation of embryos, I conducted a functional genomic screen, very much like the one that Dave Matus just talked about using RNAi. And then Melanie and Takiko in my lab took these studies forward. So what we discovered was an unexpected substrate. This was almost 10 years ago. We discovered dicer as an unexpected substrate of ERK in C. elegans germline. And dicer is very well known for its function in regulating and generating small non-coding RNAs. And it was unexpected because we were expecting a signaling molecule, for example. But this is where the power of a model organism is really illustrated, because you come up with unexpected discoveries. Dicer was unexpected for us. So we made a phosphoantibody to phosphorylated form of dicer, and we asked where is it expressed, because normally dicer functions in the cytoplasm to generate small RNAs. And the first thing we noticed here was that dicer was enriched. You're looking at phosphorylated dicer in green. And what you start to notice is dicer is enriched, phosphoform of dicer is enriched in nuclei. And it's also in the cytoplasm, but you see the strong enrichment in the nuclei. And this was very exciting for us because this was, at that time, the first description of dicer localization to the nucleus. Since then, of course, many groups have seen this. And so we went off and asked, what does phosphorylated dicer do? So we generated lines where we mutated the phosphoforms and generated phosphoalanine versions of dicer, which can't be phosphorylated. And we found that if dicer is not phosphorylated, oocytes don't develop. So here you see these large holes in the germ cells, and these oocytes are disorganized. They pretty much just collapse onto themselves and become endomitotic. They do not go on to form any progeny. They're completely sterile. So phosphorylation of dicer is now essential to form oocytes. But what does the molecule do? What does phosphorylated form of dicer do? So we sequenced these germlines, both from unphosphorylated dicer, as well as we created a phosphomimetic form of dicer, which keeps phosphorylated dicer in its constitutively phosphorylated form. And what we found is if dicer is continuously phosphorylated, as shown here in this chart, where S is converted to glutamic acid, and what you start to find is basically the small RNAs, in this particular case, the endo-siRNAs, which are 21 nucleotides in C. elegans, and these are 26G form of endo-siRNAs that are germline specific, that are not generated, whereas in wild type, these 26G endo-siRNAs are generated. So we found that phosphorylation of dicer is essential to turn off the generation of these endo-siRNAs to protect maternal transcripts. And that becomes really important because right as oocytes start to mature, dicer phosphorylation oscillates. So what you see here is arrested phosphorylated form of dicer. You still see ERK activation in red, and you basically see the green in a phosphorylated form of dicer. And right as these oocytes start to undergo release from meiosis 1 arrest, both phosphorylation of ERK as well as phosphorylation of dicer is turned off. And this is where the dephosphorylated form of dicer is essential, because now endo-siRNAs are formed. And these endo-siRNAs become important because oocytes have to undergo transition to form embryos. So all of these embryos inherit maternal RNAs, as cartooned here in yellow, and these maternal RNAs are protected in the germline because the endo-siRNAs are not formed, because dicer is phosphorylated. They're now loaded into the embryo, and they target the maternal RNAs, which are important to be cleared in readiness for zygotic genome activation. So what we found was phosphorylation of dicer is essential. ERK-dependent phosphorylation of dicer is essential to coordinate formation of oocytes with oocyte to embryo reprogramming during early development. So then we asked, okay, is this event conserved? because most of the ERK signaling is conserved. And we started out by looking at the alignment between C. elegans and human. Right on top is C. elegans, right at the bottom is human. In blue, you're looking at complete identity. And these are the two phosphosites that we had identified in C. elegans. So we did an in vitro kinase assay with human dicer, and we found that with ERK, and we found that both of these residues are phosphorylated downstream by ERK in vitro. So we did what we had done in C. elegans. We made a phosphospecific antibody to the human and mouse epitopes of dicer. And the first thing we did is we looked at some cancer cell lines. In this case, my MD PhD student, Raisa, led most of this work. And here you're looking at a non-small cell lung cancer. And we use non-small cell lung cancer for these studies because non-small cell lung cancers are largely dependent on activation of KRAS oncogene. And that was the pathway we were looking at this time. And in dark brown, what you see here is phosphorylated dicer, and very much like in C. elegans, this is nuclear. And so what Raisa found was when she did the staining with phosphodicer in this non-small cell lung cancers, there was strong expression of phosphorylated dicer. So then we said, okay, what does phosphorylated dicer do to lung cancer progression? To study that, we generated mice with phosphomimetic dicer, and we crossed them into a heterozygous KRAS G12D allele. The heterozygous KRAS G12D allele basically results in formation of tumors. And what we found is when we made a double heterozygous animal between KRAS G12D and phosphomimetic dicer, what we found was that presence of phosphomimetic dicer led to progression of these tumors where they went from becoming adenomas to adenocarcinomas. And if we waited by 24 weeks, and if we waited another 10 weeks, we found that the tumors were spread to every organ by the presence of phosphorylated dicer. And here what you're looking at is in just heterozygous KRAS, you see lung tumor lesions which don't spread to other organs. Like in this case, the heart is completely uninvaded by tumors. Whereas in the double mutant, we found tumors that had invaded to every organ, including the brain, and in this case, the heart, where the heart is completely covered by these metastatic tumors. So we said, okay, what are these cells doing? How are these cells actually invading the tumors? And so we pulled out some of these primary tumor cells. We took about 25,000 primary tumor cells from both the double mutants and the single mutant mice, and we performed single cell sequencing on them to determine all the different populations of cell types that were there. And to our surprise, we found something really unexpected. So what you're looking at in this U map here, each of these gray dots is a cancer cell. And in this particular case, it's the tumor epithelial cell, which gives rise to the whole tumor, and the AT2 cells, which are the alveolar type 2 cells. And what we found was something which we did not expect. We saw expression of gastric genes in these lungs tumor cells. Gastric genes are not supposed to be expressed in lung cells. So we said, okay, maybe we made a mistake. Maybe you're not supposed to see gastric cells there. So we zoomed into these tumors. We opened up these tumors and we did some expression analysis. And now you're zoomed into these tumors. Each of the blue dots here, blue nuclei is a tumor cell. And what we see in purple is the gastric gene. And in green, you're looking at the lung marker prosurfactant, which marks the AT2 cells. And what we found were cells that basically were double positive for lung as well as gastric, suggesting that the lung cells were transdifferentiating into a gastric-like state. And more importantly, we actually identified cells that had started to lose their lung fate, but taking on the gastric fate, where they only had the gastric cells. And so these cells were now starting to transdifferentiate from lung into a GI-like state. And if I cartoon that here, what we saw was really that presence of phosphorylation of dicer in the nucleus was leading to these lung cells turning on a gastric-like fate, and in fact turning on purely gastric-like cell fates. And we saw this across the GI lineage. We saw multiple cells that had multiple different markers that had turned on. So we said, how can phosphorylation of dicer do this? Like, how do you change the fate? And one way to change the fate is to really go into the chromatin and open the chromatin, because everything in the end is controlled through our genetic material. So we performed an ATAC-seq analysis on these tumors, on the same primary tumors that you looked at in the slide previously. And what we found was that the presence of phosphorylated dicer was opening the chromatin at various loci across the genome. And one of these loci, which I'm showing here, is the same one that we saw the expression of, which is gastrokine 2. So what you see here is that gastrokine 2 is now open, suggesting that it can lead to transcription of gastrokine 2, causing the expression. And very much like we had seen in C. elegans with phosphorylated dicer, we found that the microRNAs were generated fine in these tumors. So it was really the change in the chromatin compaction by phosphorylated dicer that was causing the opening of these loci. So then we said, okay, this is what's going on in the mouse. What happens in the human tumors? So we went back to our tissue microarray from human tumors, and we looked at tumors with both low and high expression of phosphorylated dicer in the nucleus, as I'm showing you here. And we found that for high-grade tumors, for some of the high-grade tumors, we found that indeed they were basically turning on the expression of gastrokine in these lung cancer cells, very much like we had seen in the mouse tumors. So this now opens up possibilities because it suggests that we can use our phosphodicer antibody, at least to start with, the human phosphodicer antibody, at least to start with, to detect tumors that have the potential to metastasize. So these would be tumors that would turn on, so they would have the same oncogenic mutations, but they would either have expression of phospodicer or not have expression of phospodicer. And we can at least start to stratify patients and say, these patients have expression of phosphorylated dicer, so they are more likely to metastasize and invade other tissues. So we should start thinking of therapeutic interventions for them, whereas these that don't have phosphorylated dicer and won't necessarily invade. And we're starting some of those studies as phase 0 right now. And this antibody was recently patented for these studies as well. And so, and of course, with the idea that we can detect these tumors long before they actually invade distant organs. And so with that, I think what I would like to end with today is how work in C. elegans to study germline regulation can not only inform fertility in humans, but also give us insights into how work can transcend across tissues and, in fact, give us new mechanisms towards cancer progression. In this case, I just presented this for lung cancers, but we have similar data for pancreatic and melanomas as well. And with that, I would like to thank all the awesome people who did the work, in particular Raisa, when she was in the lab and now she's a resident, and all of our funding, and all of you for your attention and staying on in this talk. Thank you so very much. And I'll take questions. Sorry, I stopped sharing.
Thank you, Swathi. Do we have any questions for Swathi? Looks like we have a hand for David Builder. I think he should be unmuted now. Let me unmute. There we go. Try now.
He's still muted.
Yeah.
Oh, yeah.
Thanks a lot, Justin, for unmuting me. Swathi, can you tell us a little about your experience trying, working with collaborators to go from, your fundamental research in C. elegans to these more directly preclinical and translational models?
Yeah, thank you for the question. So I have to say, I've had a great experience, but that's partly because all of my collaborators that helped us with all the cancer work are at MD Anderson. So we built the mouse models, for example, with my chair, who is a P53 biologist, and we worked with, so I sent out a lot of emails to our internal clinical colleagues for getting tumor samples to do some of our preclinical work, and almost all of them were very open to working with us on this. I think initially there's always a little bit of hesitancy saying, okay, do you have, would this really track? So to make it tractable for people to work with us, what we did was we actually generated the antibodies to the human and the mouse epitopes, and we did the work in cell lines before I sent them the email. So I already had cell line work showing we stimulated with FGF because we see this translocation only with FGF, but not with EGF. And so I created a panel, sent it to them, and it was actually quite, it was working with colleagues has been very easy. What hasn't been is getting funding for it through NCI, but working with colleagues here has been remarkably wonderful.
I guess that's why we're here today in part. Thanks for the
I
answer.
hope so.
All right, do we have any more questions for Swathi before we move on to the discussion? Feel free to put your questions in the chat, by the way. I can read those out, or you can just raise your hand. Okay. If anybody thinks of anything that they want to target towards any of the specific speakers, feel free to speak up or put your question in the chat during the discussion. But let's get all of our speakers back on camera for this discussion section, if you don't mind. Just waiting for Dave Reiner. But yeah, thanks to all three speakers for those wonderful presentations. During this discussion, what I really want to get into is specifically the model organism that we're talking about today, C. elegans, and its use for cancer research. So I have a couple of general questions to begin that I'm going to ask at each of these webinars, and then I'll open it up to the audience. We are running a little bit late, so it would be great if we could get all the panelists to chime in on these questions, but I will always sub in for somebody who, if they have a question in the meantime. Okay, so first question, which I think was touched on during many of your talks, but maybe just it would be nice to hear you talk a little bit more about this, is what is unique about C. elegans that makes it useful as a cancer model in all of your opinions?
I would say it's where, I think the ease of being able to do unbiased screens, the beauty of being able to go in and actually look at the mechanisms, that I think really is the power in my mind. Like we would never have come across Dicer and some of those molecules we have without being able to do these unbiased screens. And I think just that's what I would say. And I'm going to let others chime in.
Yeah, I agree completely. And I would say also the iteration speed. I mean, just the generation time and the ease of editing the genome. I think it might be maybe I'm biased, the easiest organism to do CRISPR in, but I've worked on lots of organisms. And so I think that is a really game changer.
Yeah, I mean, I think those are both great points. It's driven by genetics, historically the system, and you could probably run, the NINDS could probably run the same type of seminar, different speakers, of course. I don't think we can switch it, but the truth is that the advances there have been also driven by an intensively genetic approach, but it's branching out now into far more cell biological, as you see from all of us, and so I think as the toolkit becomes more advanced, but again, the genetics drives a lot of the fluorescence tools that we have and things like that. So it all goes back, I think, to Sidney Brenner. I would also say, and maybe this is a little solipsistic, but I just there is a camaraderie or collegiality in the system that is unparalleled. And it's a large part of why I stayed after grad school, because, and I think that feeds forward into a lot of methods that develop a lot faster. But I think the other two probably introduce the most important issues.
Thanks. Oh yeah, just looking at the chat. Okay, so I have a question from Joanna in the chat that we'll break things up with. Can you comment on the availability of or challenges with C. elegans reagents and resources? Is the community collaborative? So I guess that Dave just answered that part of the question, but maybe answer the first part.
Very.
Yeah.
Yeah.
Yeah, it's extremely, like, to give you an example, when we were working with worms, we came across this interesting result that phosphorylated dicer did not generate microRNAs. And I was like, oh my God, can't be real. I wrote to Andy Fire. I had never met him. I sent him an email. I said, can you work with us? Can you verify this data? We've been collaborators for 12 years. So it's a very collaborative field.
And then for resources, like Wormbase might be one of the best maintained, thanks to efforts of Paul Sternberg and HHMI, like the best maintained genome sort of centered database. And then the Stock Center at University of Minnesota, I mean, strains are like $10, right? Like it's amazing.
And the last bit I'll add is Dave Matus and I and there are 3 other PIs, a 5 PI paper to build a tissue-specific Degron technology for elegans. And so people just get together to get stuff done. And that paper came out remarkably fast. I should add Jordan Ward at UC Santa Cruz was the ringleader. But it's a very collaborative field. And I've found typically I'm not too shy to share things in public. People are not going to hop on your project and scoop it and things like that. So another reason I stayed in the field. I think I'm the only one here that was raised in elegans. The other two have different backgrounds than I do, but I was doing C. elegans neuroscience from grad school.
Yeah, thanks for that question, Joanna. Checking on the chat. I don't think we have anything from the audience, so I'll go ahead and ask my second question. This one, there's a lot of, I think, obvious answers to this question, but in the spirit of Dave's metaphor about trying to fit a square peg into a round hole, what are the most important differences to keep in mind between C. elegans and human cancer, specifically thinking about C. elegans as it is a cancer model organism?
I think lack of an immune system. Because in cancers, you really have to think about immune evasion, and because one of the major hallmarks of most cancers is they figure out how to evade the immune system. And I think that's where all the checkpoint studies are going in. C. elegans doesn't have one. So most of what you're studying, even if it's intra-organ communication or intracellular communication, you're not looking at a lot of actual, adaptive immune systems.
They
So
do have innate immunity, but yes, the adaptive is the key here, and Swathi's absolutely right.
Yeah, and we should just be aware that it's hundreds of millions of years of animal evolution, right? I mean, like, we like to talk about all the genes and proteins that are conserved, but of course, and then if you count like 3-day generation time, that's a lot of evolution, nematode evolution. So, they're definitely not humans.
Yeah, absolutely.
And in the past, I've actually, in flies, for example, and David Builder, I apologize for stealing your thunder. But you're probably not going to talk about this, but I've Ross Kagan, for example, has this long history of doing fly models for cancer where they're really recapitulating some of the genetics of human cancers in Drosophila in different cell types. And so, so one thing about worm development, I'm probably the least developmental biologist of the three of us here, but we all know that worms have very hardwired, ultra-fast development, right? You get an embryo, a functioning larva in 12 hours, right? From zygote to animal that can taste, eat, move around, smell, even poop. And then, another day, or day and a half to get to adulthood. The development is so hardwired, there seems to be very little plasticity that would drive what you would normally think of as a tumor. Now, the germline, there is some, of course, there are tumorous germline mutants that just proliferate out of control. I don't think they're conventional cancers. So we don't have that model. I was actually going to ask Matus if you wanted to try some of that stuff like Ross Kagan had done in flies. Like, if you get a situation where you've broken down that barrier between proliferation and invasion, can you start introducing mutants? That's one thing that occurred to me during your talk. But in elegans, typically, we don't think of it that way. I don't know if one of my more developmental colleagues want to chime in with that one.
We are a little bit pressed for time, so I want to make sure that I get this last question in from the chat. So Cheryl asks, question for Dr. Reiner: the worms that doubled their lifespan, was RAS-induced senescence involved? And this will probably be our last question.
That was doubling lifespan from exocyst deficient lethality. So there is actually some senescence and aging stuff in worms that's phenomenal, but that's not what I was talking about. These animals are dying because they can't exocytose very well. And over time, their exocytosis is getting worse because the moms had, at least were heterozygous. But by adding activated RAS or activating RAO, we actually restored exocytosis enough to get them to adulthood where normally they were dying. So it wasn't a lifespan or senescence issue. It was not croaking because the cells are dying issue. It's toxic not to have exocytosis. That's very bad.
All right. We're at 2:15, so this is the end of our scheduled time. So I just want to say thank you for those in the audience who asked really great questions. I'm sorry I couldn't get to yours, Stefano, and for the thoughtful responses. Thanks again for the participation, and I hope that everybody learned a little bit about cancer biology research in C. elegans. In the next few months, be on the lookout for an evaluation survey in your inbox. We would love your feedback on this series, and it's always a possibility that we do more on more model organisms. So thank you so much, and have a great rest of your day.
Thank you.
Thank you, everybody.